Hello everyone! Today's lesson is going to be looking at the role of mineral ions and deficiencies in plants in order to keep them happy. So, our two goals for today are to be able to describe why nitrate, calcium and magnesium ions are all useful for keeping a plant nice and healthy. And the second part is identifying mineral ion deficiencies from observations or pictures of unhealthy plants. So, let's start off by thinking about plant nutrition. Now, there's lots and lots and lots of different ions that plants require in the right balance in order to grow properly. So things like nitrogen, phosphorus, potassium, calcium, magnesium, and many, many more. And looking at this barrel diagram on the right-hand side, what you notice that if even one nutrient isn't in the right balance, then you've seen all the water spilling out. And this represents the fact that plant growth is going to be harmed if just one essential mineral is lacking. So, let's look at what our mineral ions are used for in plants, the ones that they tell us about on the spec. So there's magnesium ions, and they're used in chlorophyll for photosynthesis. There's calcium ions, which are used in pectin, or you might know it as calcium pectate, in the cell wall and the middle lamella. And finally, there is nitrogen, and nitrogen is the main component of many, many different molecules that you'll find through animal and plant cells. So amino acids and DNA, your genetic material, are really important. You have nitrogen, but also chlorophyll again for photosynthesis. So let's look at observations of where we have this magnesium ion deficiency. And what you notice is that the leaf shape seems okay, and the leaf vein area seems alright, where you see these tracks running through. Those are the leaf veins containing xylem and phloem and sclerenchyma. But the tissue in between is not having a good day. Instead of seeing this rich green colour that we're used to, we get increasing amounts of yellow deposits. So we get yellowing between the leaf veins, sometimes with reddish brown tints. So think about what pigment that might be lacking that makes it green. Next up, we've got the calcium ion deficiency, and these plants are very obviously unhappy. What you notice is that in the new growth area in particular, so this area towards the top of the plant, the new leaves look stunted, misshapen, and a very trademark feature of a calcium deficiency is that the edges, so you notice on the strawberry plant where it looks like the very tips of the leaves are dying, they're brown, that necrosis can also happen on the fruit blossoms. It could happen on developing fruits where the edges just die. Next up, we've got nitrate deficiency. And nitrate again is a very obvious one to spot visually because those leaves go very, very yellow in colour. So we have plants that have spindly yellow all the way through, or just yellow leaves, sometimes with pink tints. Using this knowledge, what I would like you to do is look at all of these different pictures we've got showing different plants with deficiencies, and I'd like you to identify which nutrient they are deficient in. So let's look through. Our first picture, where again we've got all of this yellowing around the edges and it's not looking particularly happy, this is showing that magnesium ion deficiency. Next up, we have a plant where the leaves look a normal shape, apart from they're very small and very yellow. This shows we have a deficiency of nitrates or nitrogen. On this picture here, we've got plenty green apart from the very tips and edges of the leaves, which are necrotic, they're dying. That shows that we have a calcium ion deficiency. I'm going to skip this one for a moment and we look over at this side where it's got its matching pair over here. The veins are a healthy colour, there is green across the plant, but you have those yellowed patches away from the veins, meaning this is a magnesium ion deficiency again. And finally, you can see that they've burned or they've dyed the outside of the plant leaf, but the inside tissue seems healthy. That is a calcium ion deficiency. So let's try some questions now we've had some practice. Looking at this picture over on the right hand side, you need to describe the physical appearance of this leaf. From your knowledge of plant anatomy, next you need to explain how the pattern that you've observed compares with the distribution of xylem in this leaf. So what is going on there? Next, you need to name this green pigment that's required for photosynthesis. You need to name the mineral ions or ions that you need in order to make that pigment. And then you have a bit more of a tricky one. Using your knowledge of xylem and transport together, why were these bits affected more than these bits? Why are not all parts of the leaf affected equally? And lastly, you need to suggest a treatment for the plant that will help it to make uniformly dark green leaves. What would you need to give that plant to treat it to make the rest of the leaf a more healthy colour? Pause the video and have a look through. Okay, answer time. So for our first question, the younger leaves are going to show those dark lines across the midrib that's describing our pattern, and those dark lines are branching out. But the edges of the leaves are the palest greens. This is what you might have seen. Next up, the pattern that we've seen matches the distribution of xylem vessels through the leaves. So they follow the midline, the middle bit of the leaf, and they branch out towards the leaf edge, forming a network of veins. This is where we see our xylem. The green pigment, of course, is called chlorophyll, and the ion deficiency that we're noticing here is a deficiency in magnesium ions. So, let's have a look at the next question. So for question 5, let's start with our first point. Here we have our leaf. And we know that xylem is going to carry magnesium ions to the leaves. So there's all the magnesium in the xylem. The next thing that we know happens is that magnesium ions are used to make molecules of chlorophyll. So when this magnesium is going to diffuse through the xylem into the plant tissues, you notice that you'll get the greener area closer to those xylem tissues because there's lots of magnesium available, but it couldn't get transported all the way up to the edges of the leaves. So not as much chlorophyll is made, and they will have this sort of increasingly yellowy or light green colour. So this explains the link between transport, or diffusion in this case, of magnesium ions across that leaf and the colour that you'll see the tissue. So the more magnesium, the more chlorophyll is made, and so it's a darker green, and the further away from the xylem, the less magnesium is present, and so less chlorophyll is made, making it appear yellow. And for the last question, part B, in order to fix this solution, we just need more magnesium. So where there's a deficiency, there wasn't enough to reach the edges, but without a deficiency, if you feed that plant more magnesium ions dissolved in water, there'll be enough to go around. Mark yourself on each question, and then we have a quick summary table to do, where we need to look at all of these different ions and think what characteristics can we match up with them. So let's look through our answers. So if something has a magnesium deficiency, like in our last question, we're looking for more yellowing in the leaves away from the veins. If we've got a calcium deficiency, we've got new growth with particular necrosis of the leaf or the blossom edges. And if something has a nitrogen deficiency, we might notice that the whole leaf is yellow instead and much smaller. And this leads us nicely to how we would investigate this in college. So, Core Practical 7 with plant mineral deficiencies. The first thing that we can do is look at this picture and ask ourselves, what is the independent variable, the IV, and the dependent variable, the DV? So, the independent variable is the mineral ion type that is present. So in this little investigation below, you can either have full nutrients, distilled water, phosphorus, iron, magnesium, or nitrogen. They've chosen the conditions in which to grow these plants. That's the independent variable. The dependent will be what you're measuring or what you're looking at, and in this case we're looking for the physical characteristics or properties of the plant. So let's look at how we'd plan this practical. Our aim is to investigate the effect of plant mineral deficiencies on plant growth. We've already said the IV is mineral ions and the DV is the characteristics we're observing. The equipment is standard, and you're going to have 5 solutions available. These 5 solutions will be one with all nutrients present, one that is only lacking nitrogen, one that is only lacking magnesium ions, and one that is only lacking calcium ions, and then just some distilled water that is lacking all nutrients. So we want to be able to see the effect of just this condition on plant growth, and we're going to be using these Mexican hat plants. So let's think for a moment of what other factors can affect plant growth. Because if we only want to see the effect of these solutions, we need to control everything else so that we have more validity. Take a moment thinking about plants and how they grow and think 3 things that will change that. So there's a number that you could come up with, but the main ones I'd expect you to be thinking of are the species of plant that you're using, the size of the plant that you start with, the time growing that you leave them for. You might mention the light exposure or just the volume of the mineral ion solution that you apply to each condition. So let's think about why this would happen or how we would control them. Some of them are very straightforward, some you might need to think about adapting your method for it. So take a moment to pause again and explain for each of these how you would control them. Let's start with the species of plant. So this is very straightforward. Choose the same plant species. It's really useful if you have a plant that produces many little clones of itself, like a Mexican hat plant from Bryophyllum. So each little plantlet is from the same plant and also of the same species. Very, very handy. Next up, choosing the same size plantlets, or at least ones that measure approximately the same amount for each condition and each solution. That would be very useful, because if they're all starting at the same size or a similar size, it will be easier to compare them when they've grown to see the effect. Next up, you would be sensible to give them all the same duration of time to grow, so at least 7 days to get an easily observable difference, but as long as it's the same in each condition, we're good. Next, for light exposure. Now, you cannot control the physical position of the sun, but you can at least choose where you put your plants. So if you put all your plants in the same area, so preferably a windowsill, because it doesn't have light exposure that varies that much compared to if you put them in different conditions, or if you really wanted to control it, you could set up a lamp and put a timer on so that the plants are exposed to the same amount of light through the whole time period that you're running the experiment. And lastly, to control the volume of the solution, you could measure out the same initial volume of mineral ion solutions and make sure it is topped up at the same amount. So, in order to grow them, there's a very simple way that you can fit your plants inside, and this uses a bit of tin foil that you've poked a hole in so its roots can pop through, so your plantlet is sitting happily on top of the foil. You can pop it in a test tube so that it fits inside and is floating on that top part with its little roots hanging down, and you would cover the test tube in black paper to prevent algae from growing. So as it's floating in solution, this means that as the solution goes down, so will the plant with it, and that could make it a bit more useful for you if you're running it and you can't be topping up your solutions all the time. So next we need to think about the kinds of results that we'll get from this. We could talk about quantitative results, so this is numbers and measurements that you can record, or qualitative results, such as physical observations and descriptions. So think of a few things that you might measure about these plants as the experiment runs. Let's go for about 2 each. Starting with quantitative, let's think about the easiest thing to measure with a ruler, and that would be the length of the leaves or roots. Or you could count the number of new leaves or roots. In terms of qualitative data, you might look at the colour of the leaves, the shape of the leaves, or just any other observations that you notice about the solutions or the plants themselves. So for example, here is a results table that we have modelled for you. We're measuring the colour, the general appearance and recording qualitative data, but we're also recording the height of the plant and the mean root length so that we have even more observations to compare between each category. I'd like you to challenge yourself by filling in the observations and measurements that you'd expect. So, if growth was stunted by a particular mineral ion deficiency, fill it in with what you would expect to see changed. Now, the real test of if we've understood this is from using some practice exam questions about this core practical. So here we have one focusing on mineral ions in the soil on peanut plants. I've put an arrow there to say summarize the main points in a way you understand, because this can help you answer this question a bit later. So, the effect of mineral ions on the production of fruit by peanut plants was investigated. They were grown in soil containing all the mineral ions required. After 1 week, 10 of the plants were moved into soil without calcium ions. Another 10 plants were moved into soil without magnesium ions. The 10 plants were left in the original soil. After leaving plants to grow, the mean number of flowers per plant and percentage of the flowers that formed fruit were recorded. So, I personally would draw myself a little diagram just to summarize the experiment and show what's going on. Here's what they give you. They give you a nice results table. One, you've got soil that contains all minerals, soil without calcium, and soil without magnesium ions to represent the different categories. And then there's their recordings for mean numbers of flowers per plant and the percentage of flowers producing fruit. You need to devise an investigation to determine the effect of only nitrate ion concentration on the growth of young peanut plants. You have 5 marks, so think of how you would describe different things to control, how you would run your method, and what you would record. Give it a go. Okay, so part of it is highlighted for you already, the most important bit about nitrate ion concentration. So if we're looking at the effects of nitrate ion concentration, it would be reasonable to expect a range of results of different dilutions, going from, let's say, 0% nitrates up to 100% nitrates. You would need to think about how many different intervals you pop down. So how many different concentrations of nitrate would you use in order to get a valid range of results? Next up, we need to think about different ways that you're going to control variables, or what variables you would control, and we can look at an example answer over the next page. So, thinking in terms of controls first, we could say using plants of the same species, or the same variety, or the same age, any of these factors will give you a mark. We're looking for a minimum of at least 5 different nitrate concentrations in order to have enough data. Next up, you would need to describe how you're going to control an abiotic variable, such as sunlight or humidity or temperature. So over on this example side, they give you a few methods. So if you're trying to control temperature or light, think about what equipment might be to hand in order to do that. If you've talked about the pH of soil, then you could describe them all using the same pH of soil, the same volume of water, or the same presence of other mineral ions. All of these different things you could have for one of those marks. You could also say that you're going to grow them for the same period of time, or that you're going to measure a particular aspect of growth, such as the leaf length, or the diameter of the stem if you wanted to be extra fancy, just any reasonable measure of plant growth. And then you would of course mention that we're going to do repeats in order to calculate the mean values. So annoyingly, if you don't have that calculation of mean, you cannot get 4 marks. Let's look at our next question. So we've got plants needing mineral ions to ensure healthy growth. And we've been told that the optimum concentration of nitrate ions for healthy growth of this plant is thought to be about 200 parts per million. Describe how you would carry out an investigation to find the optimum concentration of nitrate ions needed for the healthy growth of this plant in a lab. So they've said it's 200 parts per million. What experiment could you do to work out the optimum? And here we need to think about what optimum means. We're not just looking for the general effect of nitrogen ions on plant growth, we're looking for the optimum. So there is some ideas there that you need to communicate, which will make it different to the last one. But what we've learnt from our last practical is that you need to mention controls quite a lot. So think about how you would do this. 5 marks, 5 points, give it a go. Let's look through our mark scheme and see if what you've written matches up with what we're needing. So again, we're looking for controls in order to make our data more comparable, so we could use genetically similar plants, so clones, cuttings, seedlings of something from the parent plant. You would make sure that you control plants to be all the same age or size at the beginning. Again, we're looking for at least 5 different nitrate concentrations. And we're looking for a sensible range that you're suggesting. So given that it's stated in the question that 200 parts per million is the optimum, you would need to be controlling, making sure that, let's say if we have 200 in the middle as a concentration, we need to make sure that we have a few different values above 200 and a few different values below 200. So in total, from these options, you need to have 5 different concentrations recommended. You should also talk about this time 2 different abiotic variables, such as temperature, pH of the soil, sunlight exposure, things like that. And we need to have an idea of how you would measure the growth again. So just like the last question, you could measure its mass, the number of leaves, the length of the roots, the height of the plant overall. You need to suggest that it could sit to grow for a number of weeks or months, and that you would do repeats at each concentration in order for you to calculate a mean from your data. For the next question, we're looking at the availability of mineral ions and how they're affected by the pH of the soil. So you have a chart below that shows the effect of soil pH on the availability of mineral ions to plants. The width of each bar indicates the availability of each mineral ion. So if it's a skinny bar, it's not available. If it's a chunky bar, it is available. You have all these different mineral ions running down the y-axis, and on the x we are looking at soil pH between 4 and 10. So 2 marks for this one. You need to use the information from the chart suggesting the optimum soil pH for healthy growth of a plant. You need to give one reason for your answer. So looking at all these different ranges available, which is the best, and justify why it's the best. 2 marks, give it a go. Okay, looking for our answers, we're thinking about optimums. We remember that barrel diagram saying that if you have a deficiency in just one mineral ion, then you have problems for plant growth. So we're looking for fat bars all the way up. So in something like, let's say 5.5, it might be starting to get bigger for all of these nutrients apart from phosphate. That means a problem for plant growth, so you can't recommend it. So the earliest pH that you could recommend, or the lowest pH, would be going up from 6.5, where all the bars are showing you've got a readily available amount of mineral ions to plants, up until about 8, because you notice there's this little narrowing over here, so any pHs within this range would be accepted by the mark scheme. And again, just to put it in words, so you have the highest availability of all mineral ions for your plant. You could list each mineral ion separately if you'd like to, but that will give you your 2 marks. And for our last question, again, using information from the chart, you need to look at it to answer this. Explain why a low soil pH could result in reduced photosynthetic activity by plants. Talk about photosynthesis. Already there is one pigment that comes straight up to mind, or there are some things we know about what photosynthesis needs in order to occur. So let's go back and look at our diagram with those low pHs. So over here, let's say from 6 to 4, in those lower pHs, what is going to be affecting the ability of a plant in order to photosynthesize? Okay, so let's have a look through. You could say one of 4 different things with this justification. You could say, because we know that chlorophyll is the pigment in photosynthesis that's needed, and we learned earlier that magnesium is going to be a key molecule in chlorophyll for photosynthesis. If it has reduced availability, you can't synthesize it. You could also mention that nitrate we know is required for overall plant growth and many different things. So if you don't have nitrate, there's less leaves. Less leaves means you've got less chlorophyll produced and less protein synthesized. All of these things will affect photosynthesis. Again, we could talk about phosphate, a different molecule, because we know that phosphate is the P in ATP or ADP or NADP if you're a second year. And the last one we could talk about is calcium. Calcium we know is used in the cell walls in the middle lamella in pectin or calcium pectate. So if we have this issue with reduced calcium, then there is reduced middle lamella, which means that roots and plant growth is inhibited. So score yourself out of all of those total marks. I hope this has been useful for you, and feel free to send me an email or a message on Teams if you're in my class. Take care and see you next time.